No problem. Pass the book.
Bigger, you. We should have it.
Gemma, you're on mute.
Welcome everybody to today's session. We are have a Let's Talk session on understanding the role of lung inflammation in IPF with Professor Adam Byrne, Professor of Respiratory Immunology at UCD School of Medicine. If you can all put your, if you can mute yourselves before Professor Byrne begins, and Professor Byrne will speak to us for about 15 to 20 minutes and deliver a lovely presentation. Afterwards, we'll have a Q&A. And as we mentioned before, this session is being recorded, and only Professor Byrne's presentation will be delivered and recorded and delivered afterwards to everybody. I'd like to hand over now to Professor Byrne to commence, if that's okay.
Hi, everybody. Thanks very much for coming to this talk and giving me the opportunity to talk about our work on IPF. First of all, I wanted to introduce myself and the type of work that we do in my lab, and tell you a little bit about our understanding of the mechanisms of lung fibrosis, so how it happens, and our approaches to trying to understand the disease a little bit more, but also trying to come up with ways that we can intervene. So I'm a basic scientist, so I'm working at very, very early stages of lung fibrosis research, trying to understand why and when and what happens. So we're several steps before, a clinical drug or something might come into the clinic, but we're working hard to move things in that direction. So I am a basic scientist, so there's only certain questions I can answer, in terms of the clinical aspects of the disease. But I'll do my best, and I hope you understand there's some things that I shouldn't answer, not being a medical professional in that sense of the word. Okay, so just tell you a little bit about where I come from and how I came to be working on IPF. Sorry, these cameras keep let's try and hide this and see. I studied, very strangely, chemistry in UCD, and then I ended up doing a PhD in Trinity College, again on mostly chemistry. But during the drugs that I was synthesizing in the lab were modulated the immune system. So I became very interested in the immune system in the lung. And then I moved to America, where I studied in Chicago. I studied allergies and anaphylaxis. And I kind of fell into IPF research when I moved to Imperial College about 12 years ago, 14 years ago now. It was really up and coming at the time. No, there wasn't very many people interested in basic IPF research. But I started working in this Margaret Turner Wark. It's now the Margaret Turner Wark Center for Fibrosing Lung Disease. I think this is the first center for fibrosing lung disease in the world. Margaret Turner Wark worked at the Royal Brompton Hospital in London. She was a trailblazer of IPF research decades ago now, but she has lots of people who she trained who are now world leaders in IPF research. So that was at Imperial College London. So I worked there, as I say, for about 10, 12 years. And then recently I've just moved back to UCD, and I've started working very closely with Cormac McCarthy, who works in St. Vincent's Hospital, understanding chronic lung diseases. He has an interest in several chronic lung diseases, but I'm particularly interested in interstitial lung diseases and IPF. So I've been here for about 18 months, and maybe we'll decline that. That's okay. So in a very broad sense, what do we study in my lab? So I'm very interested in repair in the lung. So the lungs are unique because they're an internal organ, but they're exposed to the external environment. So when you think about it, they're really one of the only internal organ that is constantly being challenged by the external environment. So sorry, Bill, I can't give you control of the screen. That's okay. So we're constantly being exposed to different things, smoke, pollution, allergens. We all know about COVID-19 and how that changed the world. Bacteria and some allergens like house dust mite. And so the lungs it's very important that we breathe, obviously. So we have these so if you take a slice of lung tissue and you cut through it and make a very thin slice of lung tissue, you can see that the walls in the airways are very thin. And this is where gas exchange takes place, oxygen crossing into the bloodstream, because you've got some lots of blood supply there, and carbon dioxide going out. So we all know about that. But what happens in lung fibrosis is if you look at a similar kind of slice, you can see in patches of the lung, so not all the lung, some of the lung looks like this in IPF. But these thin walls in certain areas of IPF have become densely thickened. And what's happening here is you've got a cell called fibroblasts, and they lay down collagen matrix. So they're involved in wound repair in other organs and skin and things. And so in the skin, obviously, it's good that these fibroblasts work well and they lay down collagen and repair things. But in the lung, this is not a good thing because it makes the diffusing distance for oxygen. So for oxygen to be able to get into the bloodstream, really, really difficult because you've got these thickened walls. So that's the kind of classic histology or the classic pattern that we see in the lungs in IPF. And as I say, it's quite patchy. Not all of the lung looks like that. We've just zoomed into this area. And this is filled with collagen matrix is what we call it. So as I say, this is when we slice through the lung like this. So these are you've got the upper airways and then you get into the smaller airways and you have these alveoli. So where we think the injury takes place in IPF is deep down in the lungs and these alveoli. But we're really trying to understand what happens in IPF. Why does this happen? So for years, it really wasn't known exactly what has happened. But we could be beginning now, as I say, there's much more interest in IPF research. And with that interest, with that scientific interest and the clinical interest, we're starting to understand the mechanisms involved a lot more. So this is a CT scan of an IPF patient, and this is from the top down. You can see the spine in the middle here. And what you see in IPF is also this kind of honeycombing, is what we call. So this kind of pattern is a classical pattern of IPF. And again, there's a comparison between IPF lung over here and healthy lung over here. Again, you can see all this thickened fibrosis. So what is going on with IPF? What do we think is happening? Well, it's a relatively rare disease. And I think diagnosis has become a lot better and there's a lot more interest in it. So the incidence seems to be going up, but I think that's because of increased awareness. So there's the data for Ireland, actually, we don't really know the numbers of IPF, really good data, high quality data to say what the incidence of IPF is in Ireland. And we know that in the UK, for instance, I guess it's one of the more comparable countries to look at. There's about 6,000 new cases annually, and there's a median survival of 3 to 5 years. That means it's around about the average. So some people have a better prognosis. So again, as I say, you have this. Again, if you cut through the lungs and you look at it, you have this diffusion of oxygen and carbon dioxide across in the interstitium. That's really just a fancy word for the lung tissue. And the scarred interstitium broadens that distance, making it very difficult. But you have this fibrosis, and these are these fibroblasts, a cartoon of these fibroblasts, laying down collagen matrix, making everything a little bit more dense there. So how do we think it happens? Well, we know that there's some evidence for a genetic association with IPF. So there are some genes that we know tend to well, higher expression, say, of a gene, increased of some genes, we know is associated with lung fibrosis. But there's also a very, very strong environmental component. So smoking, particulates, some workplace exposures we know are associated with IPF, and it's probably a mixture of some genetic susceptibility with some environmental insults. But one of the things about IPF is it seems to be that these are things that happen maybe in middle age or in earlier life, and the injury has occurred, and then we see IPF later on in life, because one of the major associations is with aging, and with a mean age of about 65 years at the stage of diagnosis when symptoms start to appear. So when we say this genetic susceptibility, there is some signs of increased cellular aging in IPF. So for instance, there's when cells divide, when the chromosomes split, there are these things associated with called telomeres. And usually if you these telomeres shorten throughout life. So a good way to tell somebody's biological age is to look at the length of their telomeres. And so you can almost draw a straight line between biological age and telomere length. But in IPF, for some reason, there's a shortening of those telomeres. So those telomeres are shortening. So there's some association. So that would have been genetic control. So that would be some kind of genetic susceptibility with a slight advanced aging at the cellular level in the lungs, which can be associated with IPF. And then you get a hit from something or other in the environment, which maybe starts off this cascade, which leads to IPF. And we know that there's limited therapeutic options, and I'm hopeful for this. There's lots of work being done. It's amazing. From when I started about 10, 15 years ago in IPF, the amount of interest, particularly from the pharmaceutical industry and biotech, really looking to try and understand disease, first of all, so they can develop new therapeutic options. And as I say, that's one of the focuses in our lab. We're first trying to understand the disease better, and secondly, trying to come up with new therapeutic options. But granted, the stuff that we do is at a very baby stage of development. So, as I say, the lungs are constantly being challenged. So every breath that you take, you're breathing in something that your lungs need to deal with, and the lungs need to stay in that kind of pristine level where you've got very small, the walls of the airways are nice and thin, so gas exchange can take place. So there must be something that's making the decision on whether to activate the immune system and deal with something that gets inhaled, or to dampen everything down, identify things that are innocuous that aren't really causing a problem, so the immune system doesn't over respond. So, and when inflammation happens, you also need to clean up afterwards, and you have repair. So there's a balance between inflammation. Inflammation can sometimes be good because it helps deal with infections and things that need to be dealt with. Sometimes repair can be good because it's about trying to get the lungs back to that healthy state. But it's a balance between the two. If you have too much repair, you have problems. You can have fibrosis. If you have too much inflammation, then that can be bad, because bacterial infections get overtake the lung. We don't want that either. What we want is a balance between repair and inflammation, so we have healthy airways. And the cell that we think is involved in making those decisions in the lung it's a type of white blood cell called a alveolar or an airway macrophage. So macrophage means macro means big, and phage means eater. So these are the cells that most of my lab focuses on, and they're about 70 to 80% of the immune cells in the airways, which live in the alveolar spaces. And they're the first ones that sees anything that we breathe in. So they make the decision. One of the things that they do is physically eat what comes in. So if a macrophage sees the bacteria, the first thing it does is eat it. It ingests it and digests it actually, and deals with the threat. So this is a fundamental process in the airways. So these cells are critically important. So we know that if those cells are dysfunctional, you can have problems in the airways. So I've been studying these cells in the context of lung fibrosis now for quite a while. So in the airways, you have, again, this balance between inflammatory and non-inflammatory or kind of suppressive cells, which dampen down the airways. But you also have a type of these macrophages, which are more aggressive, say, and can attack whatever comes in. But sometimes there's bystander damage, which can help exacerbate lung fibrosis. So you can have a tissue resident area macrophages, these are ones that live there, or you can ones that come in from the blood. This is the tissue, the interstitium, comes in from the blood, makes its way into the airways. And what we think happens in lung fibrosis is in a genetically susceptible individual, say, or somebody who has one of these genes that are associated with lung fibrosis, there's a damage. So this is the airway epithelium. These are the walls of the airways. These get damaged from a particulate or something. And what should happen is these macrophages help repair the wound. But for some reason in IPF, that doesn't happen. And the fibroblasts, which lay down the matrix, live underneath here. So you get a damage here, and when this isn't cleaned up by the macrophages, this damage can activate the fibroblasts, which are in the tissue, and that leads to collagen being laid down. So I'm hooked. There are people who study the epithelium, these cells here. There are people who study the fibroblasts underneath, and there are people like me who study these airway macrophages. But we need to know all these three things obviously talk to each other. And so I work very closely. I had been working very closely, and I still do, with the Brompton Hospital in London as one of the biggest centres in the world for interstitial lung disease and IPF research. So I think it's really, really important that when we study the disease, we study primary cells. So these are cells that come directly from lungs, because you can model these things in animals, you can model them in cell lines that you might buy, but they're never the same as what happens in the actual lungs. So when I think when the most meaningful discoveries happen, they come from samples from the unique environment, which is the lungs. So we get samples. When I was in London, I got a lot of samples from IPF patients. So these would be patients undergoing a diagnostic bronchoscopy. So some centers use a bronchoalveolar lavage or bronchoscopy, so washing out the airways and taking the fluid from that, this BAL that we would get here. So using that to diagnose IPF. And I do that in London, but a lot of centers don't do that. They think it might be a little bit more invasive. So there's a whole discussion on whether BAL is good for diagnosis. But in London, they do. So we would get a lot of samples from bronchoalveolar lavage from IPF patients. So this would be at the stage of diagnosis. So we would also get other interstitial lung diseases. So there's a lot of related interstitial lung diseases that I also study. One of the major ones being hypersensitive pneumonitis, which is closely related to IPF. So we get samples from those, and most of the cells that you get from these are macrophages, so the cells that I'm interested in. So it just so happens that there's about 70 or 80% of the cells that we wash out from the lavage is IPF. And we need something to compare that to, so we compare it to other chronic lung diseases, but we also compare it to healthy controls, healthy volunteers. And since I've come back to Dublin now, I've been working very closely with St. Vincent's and Cormac McCarthy, and we've been getting these cells from IPF patients, as well as blood samples. The blood is very important because the cells in the circulation go into the airways. So we need to study what's in the blood. Also, we want to discover new biomarkers for prediction of disease. And the easiest thing will be a blood test. So we study the blood as well from IPF patients or healthy controls, and bronchoalveolar lavage. Sometimes we get rushes from inside the airways. These are all the epithelial cells I was talking about. They're the cells that get damaged when something is inhaled. So we want to look at the macrophages and the bronchial brushings. And then we also get sometimes when a biopsy is taken, we can embed them and we can make slices and we can look at the different things. And these are some of the techniques that we use. I'm not going to really go into those, but we can do things like if we get a live piece of tissue, we can slice it and keep it alive in the lab, where we can challenge it with different drugs and things to see if we can come up with new therapies. And this is a really good technique to use a very complex, it's non-invasive, and we can model the structures of the lung in the lab. So it needs to be a complex environment because it is a complex environment. So we need to be able to study that and how the different cells are talking to each other. So how do we measure the immune system? What do we do with these samples? So we get these samples for the bronchoalveolar valve from bronchoscopy and from blood. We can measure and quantify all the immune cells. We're very good at that. We've been doing that for years using lasers. It's called flow cytometry. So we can get really, really detailed picture of the cells that are in the blood and the airways with this technique. We can do techniques ex vivo, or in the lab. For instance, it's important that macrophages can move to get to what they want to eat or to attack. So we can compare them. So this is a chip. This is a tiny chip. Each dot here is a cell. I've put a colored dot over each cell. And I've put something that calls them across on this other side of the chip, and we can watch how fast they go. So we can say, for instance, see whether they go with purpose, or do they get stuck, or do they go in the wrong direction? And this is very important to see whether the immune system is functioning well in the lung. So we can look at this and we can compare IPF to say healthy, or COPD, or to hypersensitive pneumonitis, and see if there's a problem with phagocytosis or movement of the cells in lung fibrosis. We can stain different components of the lung. We can do these beautiful pictures, which are really meaningful to us as immunologists to understand which cells are talking to each other. And we can stain things and watch them happen in real time. So here I've stained the DNA of cells, and some cells spit out their DNA as a net. Sometimes if macrophages want to eat something and it's too big, they can spit out a net and to capture things. It's an amazing thing that's just recently been discovered. It's a very important process. So here I've challenged the cells, have added something in, and you see the color coming, that's nets being thrown out to capture the particles that I've put in. You can see these cells throwing out these nets of DNA to capture what they're doing. So we can do these very interesting experiments to look at very fundamental processes in the airways in really, really granular detail to try and understand, is the immune system functioning the way it should be during lung fibrosis? So with these techniques, what types of work are we doing? So I just wanted to really top line some of the projects that we've done recently. And so we're very interested in new predictors of the disease. So can we come up with a blood test, for example, or can we measure something in the bronchoscopy, the fluid from a bronchoscopy, that might predict IPF and how it might progress? So this is one protein that we've measured called CSF1R, and that comes from macrophages. It gets chopped off the surface of macrophages, and it's a good surrogate for macrophage number. And we found that when there was more of this protein in the airways, these were patients that didn't do as well as those that had lower amounts of it. So this is something that all these things, we're the first to discover these. When we publish them, they need to be repeated in different labs and turned into a test, hopefully. And these things take 5 to 10, 20 years sometimes. But these are things that we've discovered that we think could be useful to the scientific field and to IPF patients eventually, hopefully. So hopefully this can be validated in another lab. These things need to be repeated to make sure they're accurate. And when I was in the UK, I was very well funded by Asthma and Lung UK. They were a great organization, and they really liked some of the work that we're doing on IPF, because when I started, as I said, a lot of people were doing that. And we're interested in some of the metabolites. So how macrophages fuel their responses is very important. And sometimes they might take in glucose or sugar or fats to fuel what they do, and they spit out metabolites after they've chewed up these nutrients. And one that we found was itaconate. It was a very interesting metabolite. We found that it's anti-fibrotic. So it's spat out by macrophages only. We found that it's reduced in patients with IPF in the airways. And if we put it back in, if we put it onto fibroblasts, it stops them in their tracks and stops them from making this collagen matrix. So we're very excited about this. We're using this again, we're at the very early stages, but we're trying to improve this antifibrotic activity, and we're hoping to move that towards the clinic at some point. But this is probably our most advanced molecule that we've been talking about, we've been coming up with in terms of therapeutics. We've also found that things like iron, so iron is in pollution and it accumulates in the lungs of in some patients with IPF. Macrophages' job is to eat up some of this iron, and we found that macrophages in IPF don't do this very well. So we're trying to figure out how can you restore that function so macrophages are able to eat up this iron and protect the lungs, because when iron is free, when it hasn't been taken up by the macrophages, it can cause damage to the epithelium, but also can feed the microbiome. So you can end up with bacteria and growing that shouldn't, because they can feed on iron. So there's really a competition between the macrophages and the bacteria to get that iron. So this is only in some patients have it, and we're trying to figure out why this happens in some patients and not in others. This was funded by the Wellcome Trust, so my lab is mostly funded by the Wellcome Trust right now. And one of the major projects is really trying to figure out how we can turn an angry macrophage into a healthy macrophage. Again, by switching the metabolism, and these cells can be switch off bad metabolism, if you like. And the reason why this is quite exciting is because there's loads of drugs have been developed to do this in other diseases. And we're hoping if we understand how these macrophages become inflammatory, we can repurpose some of these drugs that are already, say, FDA approved for other conditions. Like, can we use that to change the macrophage into a more kind of happy quiescent macrophage, which dampens down that inflammation that happens in the airways during IPF. And the other thing, lastly, I didn't even know that interstitial lung disease could happen in children. I was always told, even by the consultants in the Brompton, that it doesn't really happen in IPF. And then lo and behold, I got a phone call from my sister-in-law to say that my nephew was having problems breathing. So it turned out he had a children's interstitial lung disease, which is hugely rare. And it happily turned out to be knee high. It's called neuroendocrine hyperplasia in infancy, but it's one of the diseases that children grow out of. But these babies have problems when they're very young. They puff and they find it hard to breathe. And because they find it hard to breathe, they find it hard to eat and they don't thrive. So I was really surprised when I tried to, of course, my relatives were all trying to say, well, you work on these diseases, don't you know what's going on? I was very surprised to learn that virtually nobody knows anything about these diseases. There's very little research being done. So I've been funded by Asthma and Lung UK to do some preliminary experiments just to map the immune system during this children's interstitial lung disease, using 20-year-old biopsies that have been in the basement of the Brompton Hospital, but we can do modern techniques to really to resurrect that tissue and to understand the genetics of the disease. And also we're getting patients who have grown out of the disease to come back and give blood. And then we're also collecting new bloods from babies in the Brompton. We've collected quite a few of those already. So I'm hoping that project has got legs, because I think it's a disease that really needs to be looked at as well. So thank you for listening. That's all I'm going to talk about. Hopefully I haven't thrown too much information at you, but I really also want to get some feedback, because it's really important that we include patients in our research. I know that the work that we do is very early, but in terms of the pipeline for helping patients with IPF. But it's really important, I feel, that this work is done, and we really rely on buy-in from patients, because, as I say, it's so important that we use the tissues from the site of injury and the site of disease so we can understand the disease. So I have questions for you, like, is there lived experience that could help improve our research focus? Or have I got assumptions that I shouldn't have? And I'm happy to take correction on that to improve my research in my lab. I know that the current treatments have side effects, and I'd like to hear about your experiences with those. But things like what matters most to you. Am I going the wrong track? Should I give up this and I should be researching something else, or have I overlooked anything? And again, as I say, we're hugely dependent on patients and their goodwill and their consent to help us with our studies. So if there are things that can be done better when you go to a hospital and you're asked to consent to donate tissues and blood or bronchoscopy, and it's a ways that that could be done better. And if you can suggest any new ideas or questions we haven't thought of, that'd be great too. So thanks very much for listening, and I'm happy to listen to you as well and answer any questions I can.